Thanks for the nice introduction. As an overview for today, I'll be presenting some details about the microfluidics products that will feed into the second presentation that will be presented by Juston. So as an overview for microfluidics, since I recognize it may not be a familiar term for many of you, I'll be providing a basic overview of how standard biotools, genomics products, and microfluidics contribute to the testing being performed in TruGraf. Also getting into how microfluidics compares with traditional microwell plates and the benefits provided to the end user, the opportunities for automation of our microfluidics, as well as workflow and next generation sequencing library prep, and then finally concluding with supported applications. So giving an overview of the various products within the Standard BioTools portfolio, we'll start with our instrument systems. And you can see on this slide, we're showing you the Juno on the left, Biomark HD and C1 system. Those are respectively used for NGS library prep. The Biomark HD is used for our powerhouse for genomic PCR-based applications. And then C1 is used for single cell applications. On the far right, I'm showing a picture of Biomark X, which is the latest generation of our Biomark series. It was released earlier this year, and I'll be providing a little bit more on that later in this slide deck. Moving into the next segment, we've got our integrated fluidic circuits, which are also referred to as IFCs, or perhaps you might hear them referred to as chips. These enable processing of tens to thousands of instrument-controlled nanoscale reactions per run. And I'll be providing a bit more detail on this in an upcoming slide as well. We have our consumables with the goal of providing a complete validated product bundle or kit for use by our customers. And then finally, for those who are doing custom applications, we do have a custom service whereby you can contract with them. It's a no-charge service, and they can work with you to design assays based on the needs of your applications. I mentioned the IFC, which may not be a term many of you are familiar with, and it is a foundational technology that enables Standard BioTools to automate various genomic applications at nanoliter scale. So some of the features of our IFCs are they perform multiple PCR-based applications, including targeted NGS library preparation. They can scale sample throughput without changing technologies. And by that, I mean we have different formats of IFC. So depending on what sample volumes you may have today, as needs change, you could always pivot to one of our other IFC formats to either increase or decrease your volumes. We can achieve lower cost and higher operating efficiency through volume miniaturization and workflow automation. So basically, once the IFC has been loaded with the samples and the assays, it's a load and go situation. They're designed for the ability to add, remove, replace, and replicate assays on demand. So since we're not operating in a multiplex format, each assay sample is its own reaction chamber, and they're controlled independently. So you don't have the challenges of a multiplex reaction. It's more high content without the compromise of multiplex, since none of the assays are prefixed on the IFC. And then finally, it is automation compatible. We are designed to work with upfront lab automation systems. So for high throughput labs, they can automate the delivery to the IFCs. Some of the key features and benefits of the IFC design are showcased in this slide. You can see the dimensions in the center. Those are basically the same dimensions as a microwell plate. So it's designed to have the same physical footprint as a microwell plate. They are gas permeable, so the elastomer composition prevents microscopic gas bubbles that could impact results from getting trapped in the channels. They're automation friendly. As I mentioned, they can be used with various automation lab systems. Because they are the same size as microwell plates. So anything that can process a microwell plate can process our IFCs. They're optically clear so that it makes it possible to view what's happening within the reaction chambers. We've got our R&D team that routinely sets up microscopes, and you can easily see what's happening within the individual reaction chambers if you needed to do so. They're also flexible with an open architecture that allows users to determine the order and frequency in which samples and assays are loaded onto the device. As I mentioned earlier, you can easily change, add, or remove assays as needed over time. They utilize microfluidic engineering expertise. So we do have engineers in-house that have a specific expertise in microfluidics. and that lends itself to development of different IFC formats that can control thousands of valve chambers to deliver the performance that's needed for any particular IFC. They are economical. Since you're working at the nanoliter scale, reactions are set up using less reagent and sample than traditional microwells, which reduces both waste and cost. And then finally, they are precise. Since these are controlled through our instrumentation, we provide scripts that ensure metered flow of reagents and samples to specific reaction chambers that offer a more controlled environment than microtiter plates. I'll go through this slide fairly quickly. Just another view of the center part. I wanted to focus on the reaction chambers in the middle. Since you could see the sample inlets and the assay inlets in previous slides, I've kind of expanded the scale so you can see the reaction chamber showing where an individual reaction chamber gets input from one sample inlet as well as one assay inlet. And there are thousands of these within the matrix in the center of the IFC. And this gives us it allows for combinatorial processing within each independent reaction chamber, and it allows up to 100-fold lower volumes because we're working at the nanoliter scale. A common comparison with IFCs is the microwell plate format. And so on the left, you're seeing a traditional microwell format where you've got your assay mix and your sample master mix, and you deliver each of those to the appropriate well of the plate. And as a result, each sample is interrogated only by the targets within the same well. So it is possible to multiplex, and it's not uncommon to do 4, 5, maybe even 6 different targets within a given well. But as you start getting into higher multiplexes, that drives you to have multiple wells allocated to a given sample. So for each well, that requires additional sample volume, additional reagent material to complete the reaction, and it can add to the cost. By comparison, the IFC-based PCR format, you've got the same assay mixes and the same sample mixes, but everything is independent. You don't have any multiplexing going on, as I mentioned earlier. Each sample is interrogated by every assay within the independent reaction chambers. So you get the benefit of a lot of data without the challenges of a multiplex. So some of the advantages of our format, they do support up to 192 targets per sample. They facilitate workflow automation, as I mentioned earlier, minimize the use of expensive enzymes and master mixes, conserve usage of precious samples. They're easy to optimize multiple targets since everything's independently allocated throughout the plate. It's easy to add, remove, change, and replicate assays, and they are designed to maximize throughput based on which format of IFC you employ for a given application. So kind of focusing on the simplicity of adding, changing assays. Sometimes it's easier to see that in reality versus just hearing it in spoken words. So on this slide, it's designed to kind of show you the assay inlets on the right. And in this case, there are 48 open inlets, of which I've utilized 45 of them. You can see the 3 on the bottom are empty. We routinely do this and recommend this to users to leave a few open inlets in the end so that over time, if you ever needed to add something, you can add additional assays, or in the case of this purple in the middle of the field, we've changed out an assay. We took out the original blue assay and replaced it with a pink assay. And by nature of showing this change didn't affect any of the other assays in the matrix. Everything's independent. So by changing out one assay or adding in one assay, you're only making a change to the inlet in which that change occurs. The other key benefit I mentioned is a cost one with our IFCs relative to microtiter plates. And we've had a number of customers, especially in the high throughput settings, where they've shown how much data you can generate with microfluidics relative to a microwell plate. So if you focus on the table on the bottom of the screen and look at the 2 parameters, PCR reaction volume and the number of reactions, our IFCs operate at the nanoliter scale versus the microliter scale for microtiter plates. And we have up to 9,216 independent reactions, whereas a 96-well plate is limited to 96. And the result is you end up having a case where you can generate as much data in a single IFC as you would in 96 well plates, or the same amount as 24 384 well plates. So over time, you can see it can take a while to run 96 well plates. So you're also saving not only on reagents, but you're saving on time. We've got a variety of formats available for IFCs, starting with our most simple on the far left that are basically designed for reaction partitioning. And if you progress from left to right, they've become more sophisticated over time, putting more of the workflow onto the IFC itself. So some of our more recent designs, they are designed to both capture the target that you're interested in. There are multi-step reactions that occur on the IFC itself, such as preamplification or RT-PCR. And then in the case of library prep, they're designed so that once the amplicons are barcoded on the IFC, you can harvest all your samples into a single tube because everything's been barcoded, and you can now tell the samples apart as you progress downstream to next generation sequencing. For our systems, we have the Juno system shown at the top, which is considered a preparatory system. It's designed for both library preparation for next generation sequencing, but it also processes all of our IFC formats. So it's deemed a universal controller. And it is capable of an overnight operation. You can program it to set up a run overnight so that if you wanted to do one run during a normal workday, and then you've concluded that run and the instrument is now free, you could start a run, let it run overnight while you're away, and then program it to be available when you're ready to come back the next day. So that's one of the features that I think a lot of our customers enjoy using. From Juno, you can go into two directions. You can either go into next generation sequencing. So our systems and designs are compatible with all of the Illumina platforms. We can do, in addition to targeted DNA sequencing, we can do RNA-seq from total RNA using polyadenylated targets. And then on the right, if you're kind of going down the PCR route, you would go to Biomark HD for gene expression or real-time PCR, quantitative PCR, genotyping, which is more of a qualitative result, sample ID, copy number variation testing, as well as digital PCR. I mentioned the Biomark HD early on, and I showed you a picture of Biomark X in a small little thumbprint. I wanted to take a moment to introduce our latest in the series of Biomark platforms called Biomark X. It sets a new standard for automated real-time PCR to maximize your lab proficiency or productivity, as well as with minimal effort and training, since it's designed with the touch screen so that it's easy to use and walk up to and start running from day one. It supports the same applications with a smaller footprint, allowing you to generate a maximum data output. By smaller footprint, this system is the size, it's 1/6 the size of the predecessor. And as you can see, it's a single system. It does not require a separate controller. It does not require a separate computer. Everything is built into this one platform, which is about the size of a desktop, of an old-fashioned desktop computer. It's also integrated. All the systems are now in one, and it has an intuitive user interface to make it easy for the operator to engage the system at any given time. And it's intuitive, as I mentioned, with the user interface designed for modern technologies. And it has modern components to make use of what's been developed over the last 5 to 10 years in the industry. The Biomark X is part of our overall streamlined workflow, where we now start by preparing master mixes and samples and detection assays up front, loading those onto the IFC itself, take that loaded IFC, deliver it or transfer it onto the Biomark X for cycling and imaging the IFC when the final results are available. And then those final results can be sent to any office computer for analysis using our different software packages. The overall workflow, as you can see on the slide, is about 2 to 6 hours, depending on the workflow and depending on which IFC format you're using. But in all cases, for genotyping and gene expression, the workflow involves about 30 minutes of hands-on time. So most of the operation is automated. We are designing our applications to cover various areas with the various tools that we provide. So starting from the upper left, we do offer targeted DNA-seq library assays, as well as gene expression or real-time. It's also referred to as real-time applications. We offer genotyping applications, molecular sample ID. We have a diagnostic product for SARS-CoV-2 that is under the FDA's EUA IVD, the emergency use authorization. We have products that are used for copy number variation, digital PCR, single cell analysis, RNA sequencing, and more are in development to make the Biomark series a very comprehensive and capable system in the lab. My final slide is basically just to show you the various applications that either we have developed as standard biotools or have been developed by our customers using our products. Starting with cancer investigation, looking at somatic variants in cancer or gene expression signatures to guide treatment. We've got customers who have done this for KRAS in colon cancer. And there are also other customers are looking at solid tumor driver analysis as well as immuno-oncology markers. We offer screening more on the germline variants for inherited disease. An example of this is cystic fibrosis testing, looking at the CFTR variants associated with cystic fibrosis. We offer translational and clinical research applications for genotyping correlations with disease phenotype. This would, as an example, would be BRCA variants in ovarian cancer. And then scrolling all the way down the line, you can see we've got products for pharmacogenomics, pathogen detection, microbiome testing, surveillance and food supply. Crop and ag bio is a key market for us. We've got a variety of customers using our products in novel ways there. And then finally in transplantation, which brings me to the close of this session and is a nice segue over to Juston, who will present the next portion of today's webinar.
So thank you, Bill, for the introduction, and thank you for handing it over. As you know, my name is Juston Weems. I'm the Director of Research and Development, and also the Director of the Laboratory Operations for Transplant Genomics, a division of Eurofins. So we'd like to start today and talk a little bit about what the presentation outline will look like. So we are Eurofins Transplant Genomics, but the question is, who are we and how are we relevant to whatever space that we're talking about today in biomarkers? We'll also talk a little bit about what is rejection, why is it important, and how are we utilizing gene expression profile testing to address the question of rejection. Then we'll touch significantly on the conversion from the Biomark Ph.D. and the PCR, and then finally the effect on what our biomarker testing is called with TruGraf and Omnigraph, and we will go from there. So again, we are a division of Eurofins, a large conglomerate that's an international corporation that has a number of business units across the United States. And particularly, we are part of a group of Eurofins Transplant Diagnostics, and we make up 3 different groups. We have some pre-transplant testing, which is donor screening, HLA testing, and microbiology testing that goes from a company that used to be VRL, but now is DPT, or donor product testing. Secondly, there's infectious disease that is done by a sister corporation as well, Eurofins Viracor. And then we are within rejection testing and treatment optimization and post-transplant testing. And so we are Transplant Genomics, sometimes called TGI for short. So our mission at Transplant Genomics as a personalized diagnostic company is really to be committed to organ transplant outcomes and improving those outcomes worldwide through innovative tests. And our tests are designed to detect early signs of graft injury, to differentiate among actionable causes, and really to allow for the optimization of therapy and immunosuppression. So what is rejection and why is rejection important to us? So within the idea right now is that there's a spectrum of immune activation and rejection following kidney transplantation. It's important to understand that every time an organ is transplanted within a patient, that organ is trending towards rejection. And the reason is because the host or the recipient does not recognize that is a life-saving organ. What it recognizes is that it's a foreign invader in their body. And how it reacts and how the immune system in that situation reacts is really indicative of what's going on under the surface. So here we have time of transplant and over a course of time, where you can see a couple of different things. There's towards the top, you can see protocol and surveillance biopsies in the green area. There's also an area that says biopsy indication. And then those are biopsies that are done for cause. So protocol or surveillance biopsies are basically routine biopsies that are done without any clinical suspicion of rejection. And so they're just looked at simply to see what is going on at specific intervals of time. Indication or for cause biopsies are for a change in something like creatinine or EGFR and a number of different ways where there's clinical suspicion of what's going on. So there's two types of rejection that we concern ourselves with within this context for kidney transplantation. One is subclinical acute rejection, or sub-AR, and the other one is clinical acute rejection, or CAR. And what the difference is between the two of them is subclinical rejection, or sub-AR, is not going to have any changes in serum creatinine, any changes in eGFR. And this is considered to be sort of a silent rejection that we have going on. So you can kind of see from the graph that there's inflammation that's going to flow from the sub-AR that is ultimately can, if untreated, lead to CAR, where the clinical rejection is taking place, and the for-cause biopsies are indicating that. But these subclinical rejections have traditionally only been caught through surveillance biopsy because the patients are stable and there's no outward signs of what's going on. Importantly, as these rejection events go on, chronic injury and fibrosis, or IFTA, builds up over time, and this leads in part to how we're experiencing rejection and how rejection is thought of. So how do we expand our view about what we do? So again, you have a graph that is really agnostic to time. You have course progression, and then you have a couple of different things. The first thing we think about is this transcriptomic alteration, or transcriptomic window. This is a gene profile indicating that the genes have changed, and the genes eventually will manifest into a morphological alteration where the protocol biopsy window might be able to detect subclinical rejection, or IFTA. Of course, then after that, there can be clinical alteration where this clinical window is indicated by biopsy, and that could be proteinuria, it could be GFR loss, rejection, IFTA, and elevation of serum creatinine. So when we think about transcriptomic, morphological, and clinical alterations, it's important to understand what TruGraf and what TGI is trying to do and where we're trying to approach this problem. So really, this starts with the idea of transcriptomics, and that you have DNA that gets transcribed into RNA, and that RNA gets translated into protein. Ultimately, what tends to happen is that we have DNA changes that end up in RNA changes, and then it takes a little while for the protein to catch up. The protein then will cause the structural and functional damage to the kidney that is going to be detected after biopsy or during a biopsy. So it's important to understand this. At TGI, the gene expression test that we're looking at is actually capturing at the RNA level. And we're capturing at the RNA level to see if we can figure out and understand the mechanism of rejection prior to translation and prior to the physical damage that's already occurring. So on the graph of injury, when we're looking at a kidney, we are exploiting two different mechanisms here. One is the RNA, of course, and then the other is going to be our other one is going to be our cell-free DNA product that is capturing injury. So here on the left-hand side, you can see transplant is where we start. The very first thing is that when a patient comes in with low serum creatinine, we expect these patients to be immune quiescent or have a calm immune system. TruGraf is measuring the gene expression changes of the alloimmune response that's detecting the subclinical rejection that's happening. Further down the line is the injury threshold for donor-derived cell-free DNA. And cell-free DNA is really an indicator of clinical acute rejection or clinical injury. So one thing is that the gene expression is actually looking at the patient or the recipient's immune response. Cell-free DNA is a measurement of the actual graft. And what happens as the graft is undergoing damage, apoptosis, and necrosis, the graft dumps out the donor-derived cell-free DNA into the bloodstream. And by taking a measurement of that ratio of how much donor-derived cell-free DNA is in the patient's bloodstream, we get an idea of how much graft loss that patient has had. So it's really a late marker of injury and after the injury has already occurred. So if we can catch the gene expression changes beforehand, that would really help us identify early rejection cases. So I've used the term gene expression profile a little bit now. And so the question is the what and the how of what gene expression is. So I think the first thing that I'll tell you a little bit about is how biomarker discovery is important. Then we'll discuss a little bit about what the previous technology was where this test was designed. And then again, hit the conversion to the Biomark HT and PCR and how that has enhanced what we've done. So one of the things is that there's an ideal pipeline for developing clinically usable and useful biomarkers in renal transplantation. So there's what's considered to be a preclinical phase, a clinical phase, and then of course a market. And so what we do within those perspective phases is look at a lot of different things. On the left-hand side, you see preclinical phase, where these are pre-exploratory studies. So a lot of the omics, transcriptomics, genomics, proteomics, looking at a lot of different things. And some of this can be basic science that's derived from candidate biomarkers. Beyond that, then we move into this clinical phase. Clinical phases are characterized in part by looking at retrospective studies where you can go back and use highly selected cohorts or unselected cohorts and look at longitudinal design to determine whether or not a biomarker was actually capturing something, not in real time, but in the retrospective analysis of that. Then we do have prospective studies and prospective analysis that kind of bridge the gap between the clinical phase and the market, where these are randomized clinical trials or controlled and designed standard of care designed to understand whether or not the test is performing in the metrics that the lab said it could perform in. The data that I'll show you today is largely in the clinical phase, and this is in the retrospective studies and analysis that we've done, in part with a clinical trial called CTOT-08 in conjunction with Northwestern University. So really understanding what this test has been prior is really big to us in understanding how we moved forward. So the first thing is that the TruGraf kidney test was currently on microarray technology prior to the launch of the Biomark switch. And so really one of the things that was hallmark about the microarray test is that this is a cumbersome test that really took about 5 to 6 days from RNA extraction all the way to analysis at the very end, characterized by these 3 overnight incubations that took a really, really long time to do that. Now, when we're talking about microarray is actually like a 3-dimensional stacked puzzle. And there are a number of gene targets that are on a plate, and you flow the RNA over the plate, and you're looking for hybridization or stickiness. And you can see these red dots. These are luminescent dots where that will light up if you get a match. Now, part of this is that each gene has multiple probes on there, but it's also sort of an indirect measurement of gene expression because you're looking for a 3-dimensional match in real time. Now, the risks of this, of course, include orientation, base pair changes, incomplete hybridization, fragmentation, and also what's called small non-coding RNA, that you could have a probe that maybe has 100 base pairs, and you might be able to match 5 to 10 base pairs at any random time, and it would prevent the actual match from being able to stick. So what this does is this was hallmarked by a lack of sensitivity. Now, microarray is a very good technology for discovery. It is very good at telling you on, off, up, down, but it doesn't tell you the nuance of how much it's on or how much it's off. So in part, what we decided to do is to look at this in a very different way. So again, we do a peripheral blood draw. We do an RNA extraction. And traditionally, we went to the left side of this schematic here where we did RNA gene identification via microarray. We had a 120 gene proprietary classifier that then puts out a phenotype that characterizes the patients as either having transplant excellent, meaning that they have immune quiescent as our negative test, or it's positive for gene activation and indicating that the genes are sort of moving in the direction of activation. Now, what we changed is moving over to the way that we identify the genes via the PCR system. So by identifying the genes around the PCR system, we've enhanced the sensitivity, and I'll share with you the metrics as we kind of navigate this shortly. So I showed you the process of the microarray being a 5-day process. Now I'll share with you this TruGraf kidney technologically improvement that we've seen with standard biotools and the Biomark HD in particular. So we prepare the samples, we do RNA extraction in a very similar way, but now we're looking at the genes in response to the 120 genes in the PCR-based system. So then we transfer to an IFC, it get processed on the Juno, which again is the liquid automated handler that you heard about earlier. And then of course the amplify and detect on the Biomark HD, which is our PCR machine, and then we analyze the data that comes out of that. So again, one of the important things is to think about how the dynamic range of sensitivity changed across the two different technologies. And so when we're thinking about the practicality of the qPCR versus the microarray methodology, and a publication of a few years ago about looking at mRNA expression using microfluidics dynamic ranges in the biomark, but also thinking about what's happening with the gene array, it kind of gives us an indicator of how this changes. So on the vertical axis is the log fold change on microarray. And I've drawn two red lines to indicate to you basically the high and the low of fold change that we're being able to see with our microarray type technology. And you can see it really goes from about 3 to about -1 fold change, which indicates there's about a 4-fold change that we're able to detect within our microarray data. Now, when we look at what happens when we move over to the biomark, suddenly we see a huge increase in that dynamic range. And it really opens up to about 13-fold difference that we're able to see in the sensitivity by going to a PCR-based system, and in particular, using microfluidics of standard biotools and what we're doing with the Biomark HD. So this ultimately is our practicality for how we're doing that. And so as we did this conversion, we wanted to think about what the data looked like and how did this enhance this. So again, just to reiterate what the gene expression profile is it's a test that looks at RNA, and it's designed to confirm quiescence. And we're looking at the gene expression profile of 2 reference populations with stable renal function. The biopsy is confirmed as either normal histology or that sub-AR, subclinical rejection. And so our test reports out a binary result, a negative test, that is an immune quiescent, and that is a normal histology, and it is clear of subclinical rejection. Traditionally reported out as TX, but we're navigating away to a negative positive for ease of use. The positive test, of course, is the not TX, and that is not transplant excidence in the case where further evaluation may be necessary, and the profile looks more similar to that of subclinical rejection. The intended use that we have within the assay here is as an alternative to surveillance biopsy in kidney transplant patients with stable renal function who are 90 days post-transplant, also in aiding and thinking about how immunosuppression medications can be optimized, and also thinking about how the kidney is doing at any given time as clinically indicated. So what does our technological evolution look like? Well, looking again to the CTOT clinical trial study and the validation samples, where we have a multiple of over 800 biopsy paired samples. On the left-hand side, we looked at microarray as a test, utilizing some bioinformatics as random forest. The test that we had gave a negative predictive value of 87%, a positive predictive value of 45%. Using now the standard BioTools PCR-based system, at one time called Fluidigm, using the exact same random forest approach, we enhanced both the positive predictive value from 45 to 65, and we enhanced the negative predictive value from 87 to 92. Now, importantly, what this has allowed us to do is not only have a more sensitive test or a more sensitive assay to actually understand it, but it actually allowed us, by using microfluidics, to reduce the amount of blood necessary and RNA necessary to process these samples for our patients. And so what we really have is 2 tests, as I told you about, a gene expression test where it's pre-injury. It's the earliest immune activation test that you're going to be able to see changes, indicating that rejection could be a possibility and that something might be brewing on the surface. TRAC, our ViraCore TRAC, is our donor-derived cell-free DNA product that I mentioned earlier, where it's sort of a late marker of renal dysfunction. And together, we've combined these 2 tests into something that we at TGI have launched as Omnigraph. Omnigraph enabling earlier, smarter clinical answers by combining complementary biomarkers sensitive to different types of rejection for a better diagnostic confidence to allow physicians to provide more coverage on the types of rejections that we have. So as I say this, the next thing is sort of to finally talk about what the effect is on the TruGraf gene expression test, and then how that changed our Omnigraph. So in data that was published in CJSON with Northwestern and a number of our collaborators, the microarray data of looking at the two different biomarker tests that we have, the cell-free DNA and the gene expression test are on the left-hand side. And then the data that's been recently published on the Journal of Transplantation and Technology Research is the data that has been done with Standard BioTools, formerly Fluidigm, indicating how these metrics have changed. So this Venn diagram is a busy slide to try to navigate. So what I'll do is just draw your eyes to a couple of different things. The first thing is on the peach side, the square, those are biopsy-proven rejection events. The bluish square on the other side are the biopsy-proven normal events. The green circle is the donor-derived cell-free DNA positives. And then the orange circle is the TruGraf gene expression positive tests that were indicated from there. So what you're seeing is you'll see on the left-hand side, there's 27, 21, and 23 respectively, indicating that's how many biopsy-proven rejection events were captured by the biomarkers and listed as our true positives. So you can see the TruGraf one alone at 23 samples. And so when we think about what happened when we changed over to the PCR-based system, is that 23 number went up to 36, indicating to us that we captured at least 13 more rejections. And again, if the goal here is to capture rejection, this is what we're enhancing by going to a more sensitive methodology within the PCR-based system. Second to that is the 45. These are the ones that the TruGraf score called a rejection event, but the biopsy disagreed and we saw no rejection in the biopsy. Sometimes those are called false positives. And so what we see there is that we reduce that number to about half. We reduce the number of what is considered false positives. One thing I would say, though, is when we think about the gold standard of biopsy, is that biopsy is actually a moving target. So it's not actually a hard and solid gold standard. And importantly, what TruGraf is capturing sometimes might happen before the biopsy is showing damage. So it's really important to think about how gene expression is related in that omics pathway that I mentioned earlier and how these things are navigating together. Finally, the thing that I'll mention is actually the false negatives. So both tests came back negative, but 32 of those tests were shown to have a biopsy-proven rejection event. Again, we reduced that number by half, and that was really, really important. But I think overall, what you can see from this Venn diagram is that when we think about biomarkers, sometimes people use metrics like NPV and PPV, sensitivity, specificity, and then think that they're comparing apples to apples. I think the most interesting thing about this data as we published this was that the overlap between the two tests, the cell-free DNA and the gene expression, is not 100%. So the question is, what is cell-free DNA doing and what is the gene expression doing, because they don't overlap? So when we looked at this more closely, we decided to drill into the idea of the type of rejections that are happening. So again, the green is going to be a gene expression profile. That's our TruGraf score. Our blue circles are going to be the cell-free DNA positives. And really, it's split into three groups. It's antibody-mediated rejection, ACR, in the left-hand corner. It's AMR. T-cell-mediated rejection in the upper left-hand corner, the antibody-mediated rejection in the right-hand corner, and a mixture of both ACR and AMR in that bottom quadrant right there. And so again, a busy slide, but let's highlight a couple of key points here. The first point here is that we're actually capturing a number of different rejections by looking at the two types of tests. You can see that from the TruGraf aspect, you went from 16 tests to 29 tests. And you can actually see that the 4 tests that overlap that are highlighted here from the cell-free DNA were all captured by our TruGraf test as well. And this is by changing over to a new technology. Second to that, you see that the 10 reduced from 19. And those were 19 tests originally that TruGraf missed. It has now been reduced to only 10 tests that TruGraf didn't capture, and it's increased the number of that overlap from 12 to 21. And finally, you can see that it's capturing a number of the mixed rejections within there. And so really, when we think about all of these rejections, there were 103 rejections that were in this cohort, and there's about 19 of them that both tests missed, but there's only 17 that the gene expression missed when we switched over to a different technology again, when we went over to the PCR-based technology. And so ultimately what you see is we reduced that number and we're capturing more rejections by using the TruGraf PCR. We did not change anything in the cell-free DNA product. And so it's really important to understand that the enhancements here that we're getting out of this are simply because we have gone from a microarray technology to this PCR microfluidics technology. So the last thing before I hit some of the other things to talk about would be that what we've done at TGI at Transplant Genomics is try to increase the biomarker diagnostic performance to a new standard. And we've utilized technology to be able to get us there. So when we first launched TruGraf microarray, you can see in the orange bar on the left-hand side that it was a 48 and 91% positive and negative predictive value respectively. The TRAC cell-free DNA has very similar performance metrics. Now, what we did in conjunction with Northwestern University is to publish data for the very first time to show how the two biomarkers can be complementary to each other. And so even though they have similar NPVs, they're ruling out different types of rejections. And so you can see with the C. Jason paper that we increased our positive predictive values from the 40s all the way to the 80s by just looking at a double positive predictive or a double negative in their predictive values. And then in the next month or two, we looked at the TruGraf PCR, getting it off of the microarray technology onto the Biomark HD and enhanced the positive predictive value just a little bit more. Now, this is what we have overall, our commercial available product now, which is the Omnigraph, where you get the TruGraf PCR plus TRAC in one tube that in one report, is now we have, when both tests indicate positive, we have a positive predictive value of 89%, almost 90%, making it the highest positive predictive testing on the market. When both tests are negative, indicating immune quiescence and no injury, we have a 94% negative predictive value. So when we think about what this has done and what this is doing for our patients, our clinicians, and getting us out there, is that by combining the test, we have created something that has a high positive and a high negative predictive value. But importantly, this change was facilitated by getting away from the lack of sensitivity that we found in microarray and onto a more sensitive, more technologically advanced solution with the Biomark HD. So now we offer this as a single 6 ml routine blood draw. So previously you had to have 3 different tubes to be able to draw multiple tests. And now we've simplified this to one simple blood draw that can be done in a normal of different ways. And also no special packaging that comes with it, no cold packs, no shipping it frozen or anything like that, with high level of stability that comes with that. Secondary to that, of course, is one easy to interpret longitudinal report that is showing you what is happening both for TruGraf and TRAC over the course of time, and hopefully providing a more granular detail for the patients and clinicians to better gauge the health of the kidney overall. So again, combining these tests, I think the biggest thing that I would say is that by looking at gene expression or looking at cell-free DNA alone, TruGraf or TRAC, you are not giving full coverage of potential rejections on both sides here. So by combining this test and offering this, we provide the value of offering 2 tests in one tube, one report that has been really, really important in navigating this moving forward. So with that, I appreciate your time and I'm happy to answer any questions that you may have.